Feast your eyes on these painted ladies, an apt name for a butterfly so vibrant.
Butterflies and moths constitute 10% of all known biodiversity in terms of numbers of species, and they really display in front of us a rich canvas of colors and patterns on their wings.
With support from the National Science Foundation, developmental biologist Arno Martin and a team at George Washington University are using cutting-edge genomic techniques to better understand how the rich stripes and swirls of a butterfly's wing take their shape.
The wing patterns are used in nature for camouflage, avoiding predators, signaling toxicity, and also to find mates. So we know why they are so diverse, but now the question, I would say, is to understand how it happens. How do you create such an explosive diversity during a deep evolutionary time?
The team uses the genome editing tool called CRISPR, which cuts DNA at targeted locations, like a pair of molecular scissors.
The way we use CRISPR here in this lab, it's simply to make little snips in the genome to modify genes, and then we see what is going wrong during butterfly development, and it's sometimes translated into some modification of the wing patterns.
They start by gathering eggs, these little blue spheres. Once they get them all lined up under the microscope, each gets a little squirt of CRISPR cocktail. They're trying to knock out what Martin calls the master paintbrush genes, ones they suspect control wing shapes and patterns.
And we say you knock it out, which is very imaginative, right? You have lots of genes in the genome, you take one that you think is important, you knock it out. And you see what happens, what is going wrong. It's like studying maybe how a car is functioning, but you break a little part and you try to understand what's wrong. Is it the wipers that are stopping functioning, the electric system, the engine, and so on?
The eggs hatch into caterpillars, which eventually morph into butterflies. They can actually watch the wing patterns take shape every step of the way. When unexpected shapes emerge, they know they found a master switch.
Sometimes the effect can be a little bit predicted based on the way the gene is expressed during wing development. When the wing is growing in the caterpillar, in the larva, the genes are already expressed in territories that are the future wing patterns. So sometimes the master switch gene we're interested in, for instance, is expressed as a stripe. And when we see that, we predict that if we remove the gene, there is something looking like this stripe that is going to be gone, and very often it has happened.
It's not all about butterflies. Variations on the same master genes are also found in the genomes of many animals, driving the development of the shapes and the colors that make each kind unique.
So this master switch, this master paintbrush gene, if you want, is of particular interest because we know that in the genome, around this gene, there is a lot of genomic change that is shaping biodiversity. We really want to dissect that at the molecular level, go after the little details that really create novelty during evolution.
Looking to butterflies to understand the genomics of biodiversity. For Science Nation, I'm Miles O'Brien.